All right, so hello everybody. I am George,
Hi, George.
and I don't enjoy intros too much, so let me just jump into it. The subject is feedback loops in biology, and more importantly, how we should change paradigms of research in biology to account for these feedback loops that keep popping up. A headline I have is like, a biological system understands itself through query action cycles. And I want to give you an intuition about this through the simple example of moving a muscle. Not a complex movement, just a few milliseconds of deciding, oh, I want to raise my hand. So, first of all, you have like nerves firing all the way through the cortex, through the cerebellum, to those muscle centers. Then neurons fire back from these group of muscles, and from the eyes, and from the vestibular system, and so on, back through your brain to the cortex. This is feedback to what the cortex just communicated, and to the movement that just happened. And then your cortex adjusts based on that feedback. So then in turn the cerebellum, as part of this rather, the cerebellum will also like filter a lot of these signals, both the ones coming in, the ones going out. And the muscles will be reporting a bunch of stuff back to the brain. For example, something that translates to position in space, concentration of DHB, lactic acid, glucose, etc. There's also feedback processes that are happening, again, during this very simple, like few millisecond movement at the level of cells. So the membrane of cells will query things like, what are the level of nutrients in the serum they're in? Figure out how to allow more or less of these nutrients in. The same happens with the nuclei of those myocytes, and the same happens with the mitochondrial membranes. The mitochondria will produce ATP, and as part of that, reactive oxygen species will be produced, and the mitochondria will have to produce various compounds to get rid of those reactive oxygen species. So now the DNA of the mitochondria folds and is being read, and is being transcribed in such a way as to produce like sufficient compounds to like have that mitochondria not die, yet produce sufficient ATP to account for the demands of the nuclei. And then the nuclei does the same thing. It clears reactive oxygen species. It has various mechanisms of like demanding a certain level of energy from the mitochondria. It has certain mechanisms of figuring out how much the mitochondria can actually output, and so on and so forth. So there's this like complex dance where every action is met with feedback to that action, and then a new action is taken to adjust based on that at many different levels. Okay, so what I'm trying to say here is not like biology is complex. I think that complexity is like a cope. What I'm trying to say here is that given that we describe these simple actions with such a level of complexity, we might be doing something wrong. If you try to describe, say, the movement of the planets by just describing snapshots of planets, then planets are complex. If you have a law of how the planets move, then planets are incredibly simple. So the movement of the planets is roughly gravity. And I think we should be able to capture the movement of a muscle approximately with a rule about as simple as gravity. Not to describe it fully, but to describe most of the relevant things going on, to be able to predict what's going to happen, to be able to influence it in an experimental setting. So yeah, the thing that stands out in this complexity soup is, again, feedback. Feedback that happens at any level for the body to do anything. Feedback without which we very, very soon have any organism, from a simple bacteria to a human, cease to function and exist. So the interesting thing about the kind of objects that physics study is not where are they, but it's something like how do they move. The primitive of physics is not objects, but it's movement. Objects are a bad abstraction that is required to express concepts about movement. And objects are nowhere near as relevant to physics as movement. objects go away much faster than movement in any physical paradigm. So similarly, I think the feedbacks of biology should probably be feedback loops. Or at least we should consider really hard if this is a good primitive for understanding biological systems. Not because feedback loops can help us build useful artifacts, and not because feedback loops are omnipresent in biology, but because both of these factors combine. If something is both useful and frequent, it's probably a good thing to describe the system that we observe it in. So let me diverge and get a bit practical and go into like medicine and a more sort of like human enhancement and transhumanist take on medicine. And look at some feedback loop devices that already exist. So one of them would be a hybrid closed loop insulin pump. This is a very simple device. You measure glucose levels and you inject insulin. Because insulin controls glucose level. If you have diabetes, you can't quite control glucose levels. So you're essentially doing what your pancreas is supposed to be doing. And while this is a very simple device, the interesting thing is we could have built it roughly 30 years before the first one was built. All the technology existed. And you didn't need any sort of like Manhattan Project. You needed about 1 or 2 guys with domain knowledge and a week to build one. And yet it took 30 years before the first prototype was built. And they're still not in mass adoption, even though they are much better than alternatives to dosing insulin. And it will take about 50 years probably, half a century, from when this device was creatable to the point where it's actually mass market. Bypass machines also use feedback loops. You have to look at the level of electrolytes in the blood, you have to look at oxygenation, you have to look at CO2 concentration, you have to look at pressure, and you have to adjust all of those in order to keep a person alive without the heart and lungs. Now to go into feedback loops that like would have a human in the loop, you have various like osteopathic techniques. Muscle energy techniques are like interesting because they essentially have a human give you feedback on what muscles you should move, and then you move those muscles in order to get rid of some sort of pain, get rid of tension, or adjust something. And this is a fairly slow feedback loop. If a, say, bypass machine has feedback loops operating at the level of milliseconds, these techniques would be at the level of minutes. And neurofeedback, which has been used in many cases successfully to reintegrate individuals in society. So take someone who's essentially non-functional, and by training them to undertake actions while looking at their own brain waves and explaining what that means, slowly teaching them a better way of being. It's usually used for individuals with poor executive function, but also to retrain people to move and so on. So feedback medicine looks something like, oh, you have a problem, you try to find a marker for what's wrong, you intervene, you see if it's still wrong, it usually is, you intervene again, and you do this maybe a few thousand or a few million times. And the generic form of a feedback device is essentially a device that adjusts its action based on a constant stream of inputs, trying to understand what, if anything, those actions are doing to affect the stream of inputs. A feedback loop intervention is not designed to solve a problem, it's designed to learn how to solve a problem. So let's look at some simple devices that don't exist. We could, for example, in this feedback paradigm, build sleeping masks that help you fall asleep by monitoring the tension of face muscles and intervening with things like temperature variations. We could be building EEG headsets with near-infrared simulation to improve things like attention. We could be building pump and measurement devices for blood coagulation that measure a coagulation index and then slowly inject some anticoagulant in order to keep coagulation levels in a very precise range. Whereas right now, dosing anticoagulants is something that you adjust maybe every week. If you have a severe condition, every day. If you're in a hospital, every 20 minutes. Feasibly, it could be done every 30 seconds without any doctor in the loop. For techniques like MET, you could have muscle tension trackers that essentially monitor the state of muscles and tell people how to move in order to like release those tensions. And to get rid of pains or to avoid injury, ideally to never have those pains. And what they really wanted to showcase with those 4 devices, when I thought up about them, was existing devices. I thought, oh, what are things that I could build in like 2 days with minimal tooling? Let me Google that, because these things surely exist. And I can make a point that some of these devices like are here and look at how they're used. And it turns out that none of those devices exist. Well, one of them exists right now, but the other 3 still don't exist. So I'll go back to the theoretical level for a second. Look back, if you may. The way change happens in biological systems is through monitored action, through action that takes feedback and adjusts itself. When we poke at biology right now, we are simply operating with the wrong primitives. Taking action without feedback is doing something that's simply not in the same problem space that biology is solving for. So a feedback loop paradigm would have at the least 2 pillars. One of them is that input-output loops, or query action loops, should be the primary building block of any experiment or theory. The second is that systems should be described by learning algorithms that are able to quickly learn mapping between those inputs and outputs. We should have no fixed equations, we should have no static pictures, or at least no complex static pictures. Nothing like a proteome or a transcriptome. Finally, like a small caveat, what I'm working on right now are feedback loop devices to improve various cognitive functions, such as the aforementioned headset. And yeah, this is a bunch of notes and references, which might be available, but really nobody reads references, so why would you? Hopefully this gives you an intuition about feedback loops and why we should probably be using them more when you think about biology and about affecting change in a way that's useful to people. That is all. Oh, and I think there's like a Q&A for people that want to ask questions.
I have a question.
Ask a question.
The device on the picture, what does it do? What is it called and what is the function? What does it measure?
It's a toy that measures surface voltages on the skin of your head, and it has this version, I think like 5 stimulators with near infrared that interfere in like specific areas. It tries to compute a measure of like focus from those surface voltages, and then it tries to affect that measure of focus using those near infrared lasers, well, not lasers, LEDs, by alternating the rate at which they pulse and which ones are activated.
So, the way I understand near infrared lights, or you said lasers, right?
No, it's LEDs, it's not lasers at all. It's actually very unfocused. And this is more of a, again, like...
But it's a red light, right?
Near infrared, yeah. Like closer to like heat than to red light.
Yeah.
Or to heat radiation.
Near infrared. So it improves like oxygenation, metabolism, things like that, right?
Right, but the core point here is precisely that the kind of devices I want to build aren't ones where I have to assume what my intervention is doing. They're just ones where I have to be able to monitor the thing I care about, or a good proxy for the thing I care about. So, for example, let's say that what I care about is concentration, and I know that certain patterns of like voltage at the surface of my skin are related to concentration, which is questionable, but like by that imperfect premise. At that point, the only thing I have to do is try different patterns of near-infrared light and figure out what kind of patterns actually affect those surface voltages in a way that I want. Now, it might be that they don't have any effect at all, and then I have to give up and try some other intervention like a transcranial ultrasound or maybe like light simulation of the eyes, or who knows. But the point is that I can be completely atheoretical about what near-infrared light is doing. I need like a hunch that it's doing something and that I might be able to find a useful way to use it.
I think it's 100% doing something, because I did use near-infrared light on my head, and I felt it was stimulating to me, like coffee.
Yeah, I also think phenomenologically that like I confirm it's doing something. Yeah. But it's harder to find like a specific way to use it for a goal, I guess. Maybe not. Like you felt that it was just like a stimulant baseline.
Yeah, it improves the blood flow and stuff like that. And, yeah. So you're trying to get a feedback of the electric current, and then basically use the near-infrared light to adjust the electric current going through your skin.
Yes, pretty much. I mean, the electric current going through my skin hopefully is a marker of what my brain is doing. So what I'm trying to do is use near-infrared light to alter some properties of the brain / the mind, which is reflected in those surface voltages. And this is, again, maybe not the most straightforward example. It's the one I'm interested in.
Those are dangerous.
Yes?
I'm curious, have you looked at simpler feedback patterns related to those brain states, such as facial expression and these kind of things? Like when you're talking about focus or specific emotional states, how those might affect or relate?
Have I looked at? Yes, I have some specific data sets placing a camera in front of the face. Right now I'm actually trying to like look at correlations there between like eye movement, muscle tension, and what the vasculature is doing insofar as it's like visible under green light. But I haven't built a device based on this. It turns out that like building electronics is hard, and I've only started building electronics like 6 months ago. So I'm very slow at this, especially since I have to do other things in parallel. This is like a weird hobby more so than a full-time thing.
You talked about like feedback between muscle to help with like ligament tangleness.
Well, about two of them. One of them being like kind of what osteopaths would do. I guess no, that was the only one, was it?
Yeah, I think so, yeah. Yeah, how exactly does that work? I don't follow, like, what action would you apply?
What action you would do?
Yeah.
Presumably either use shock waves as an action or heat would be the simplest one. Usually heat is enough to like relax or contract a muscle if applied correctly. Or you could also use literal mechanical stimulation like a massage gun style thing, though the form factor for that is more complicated.
And then how would you detect, what would the feedback be?
The feedback would be measuring tension in the area using something like, surface voltages again are like very easy here. There's not that many things that you can see at the surface of like a human skin. You can look at vasculature, you can look at surface voltages, you can look at vasculature like 4 to 8 centimeters beneath that. You can use ultrasound to get like a very rough but high frequency image of tissue. In the case of trying to help with muscle relaxation, in the best world I would probably use ultrasound. Like ultrasound tends to have some interesting properties around measuring like the stiffness of tissue, and if what I'm trying to do is like in most cases just like unstiffen an area, that's probably most useful. But I haven't thought about this problem more than like 2 minutes. No, I thought more than 2 minutes, but like not very many minutes, I guess.
Like, I'm not too familiar with how a TENS device works. So it feels like you're kind of describing a TENS device.
Yeah, so a TENS device could also work. A TENS device I find to be quite invasive. The problem with a TENS device is that it has a good action, which is essentially sending electricity through a muscle fiber in order to make it contract, but it has no feedback. So I would love a TENS device that is sending that electricity through the muscle fiber, but then it's also looking at like, oh, what's happening with this muscle as I'm sending this? So how you measure, okay, so there's two questions. How you measure executive function when manual biofeedback is being done is by essentially having the researchers sit with, usually these would be like kids with like serious disabilities. So much closer to like, oh, this kid can't speak by age 12 as opposed to like ADHD. And try to see how well they're performing on a certain task. Then trying to correlate, like the researcher then tries to correlate that with what's happening on an EEG profile, for example. And then instead of telling the kid, oh, try to read this piece of text, they tell them, try to make this shape on the EEG look in this specific way. By already knowing kind of like how that shape looks like when the kid gets what they're reading. And it usually tends to be a fairly slow process, like it takes a few months to like train people in this way, but it also has sometimes surprisingly good effects. Like the largest I have seen in literature is taking a pair of twins that were essentially like dysfunctional, so considered to be like, on a kid's IQ test they were scoring like 67 and 72, so essentially like cannot read, cannot write, can like barely-ish communicate, and essentially bring them to like normal scores and like normal performance in a half year of doing this. With adults it tends to be harder, and it's a very slow process. How you can automate this, that's like a harder question. You either have to make inferences like, oh, certain patterns on an EEG would correlate with attention, so we'll treat that as though it's attention, or you have to add more bells and whistles. So for example, your device could be tracking someone's eyes and could be tracking the environment and could be seeing the speed at which the eyes adjust to moving objects in the environment. And by measuring that reaction speed and then trying to like increase it with feedback, that's a very good proxy of something like attention. It's like, or rather something like active awareness, let's say. But yeah, doing this with feedback is like slightly more complex, so that's a bit of a problem, and you can never get it like perfect. This is roughly a core problem in medicine as a whole, that the things that you can measure exactly and accurately are usually not ultimately the things that we actually want. The best we can hope for is to achieve like imperfect proxies. But at this point we do have a lot of imperfect proxies that we could be using.